Is that a thumbs up? Whoa, yes, I guess that's a thumbs up. Hello, welcome all. I am Jamie, and this is Bioinformatic Algorithms using F#. And I will be going between PowerPoint and code. Let me apologize for my PowerPoint skills. It's not particularly strong. Better in the code. Real quick, I'm from the United States. I live in Denver, Colorado. Data scientist right now. My 9-to-5 job. My 5-to-9 job, I did a lot of community building, helping out with Visual Studio and Data Plat for Microsoft, so as an MVP. No longer doing that because I'm actually volunteering at 2 labs in the Denver, Colorado area. Wet lab, and that's what you see biologists with pipettes and Petri dishes and going through biosamples. That's the lab at CU Anschutz. And then I'm also a volunteer at the dry lab, which is data scientists like myself looking at the results of those experiments. And that's at University of Colorado at Denver, or CU Denver. Boulder, excuse me, Boulder. And I'm also a coffee farmer, so if that interests you. Real quick, this is NDC, Developer Conference. Are you all developers and then software engineers? And that's who I'm talking to, yeah? Any data scientists? I can't, no bioinformaticians? Okay, well, maybe I'll convince 1 or 2 of you to convert on over. Any coffee farmers? Any coffee? Any coffee lovers? Yes, yes. Just have you know that as the price of coffee increases, it really doesn't come to the farmers. I guess Starbucks needs their money. And today, we are going to talk about functional bioinformatic algorithms. And this is when I shifted over to start doing bioinformatics about 3 years ago. I sort of taught myself. I went to the local university, the local uni, took biology classes. I already knew the software engineering piece of it for many years. And I took some online classes. And there's this wonderful book by Computational and Peisner called Bioinformatics Algorithms. There it is. And if you are interested in this topic, I'd strongly encourage you to buy that book and work through it. It's a fantastic book. When I got to the dry lab, the layer lab, I was assigned to a project not doing DNA and RNA, which we'll talk about in a little bit, but doing mass spectrometry data, looking at proteins and breaking down proteins. So that's the code on the right. It's called Hype Search. And we're trying to cure type 1 diabetes using Hype Search, looking at hybrid peptides. So real quick, before we jump into the code, which I know you all want to do, I'm going to take about 10 minutes to show you some biology, to give you some experience in the domain. If you've taken biology in high school, this should be a fairly basic review. The central dogma, posted by, was it Watson or Crick? I can't remember which one. That DNA makes RNA, which turns around and makes proteins, and you, me, living things are collections of proteins. So I'm going to hit run on this. I found this, I studied it at the bottom, this YouTube, and I think the graphics on YouTube for a lot of these things are fantastic in the last several years, but inside every cell, which you're all made of, most cells have nucleuses. And inside there are chromosomes, which are the drawer for keeping all your DNA in order. And then when you unwrap it, you can see that it's actually, I'm going to hit pause for a second, it's a sugar phosphate backbone. That's what those blue strings are. It's double helix. And then they're joined by 4, they're joined by 4, for lack of a better word, hydrogen, hydroxyl, no, they're not hydroxyls. I can't remember their exact word, but there are 4 things, adenine, guanine, thymine, and cytosine. And in a DNA, they always pair together because it's a double helix. So this red here might be an A, and then that would then be a T, the green. And then over here, this would be a C, and that always pairs with a G. So if you know one side of a DNA, you certainly know the other side too, because they always pair. Inside your DNA, which you have about 4 billion of those base pairs, there are certain sections, the gene region, and that actually turns around. And a certain enzyme called RNA polymerase takes it, splits it apart, takes all these free molecules, joins them together, and makes RNA. The only real difference is that T is replaced by U, and that becomes messenger RNA. One thing, and that's something that affects us in my lab, we're very much interested in, is that RNA then gets cut, spliced, moved around a little bit, some post-processing, as you were. gets kicked out of the nucleus, this messenger RNA, and then there's yet another enzyme coming along, the ribosome, and that's going to then take the free other pieces, another kind of RNA that carries an amino acid, and notice three of the pieces, and they match on, and then you'll see that the amino acids chain together, and after a certain point, the amino acids stop chaining together, and you have it folds together, and it turns into protein. You do enough at scale, and you have you and me. So that's the central dogma, and that's how it all works. This is important to know because we are going to be working with DNA. There's another really cool video that I want to show. Here, no, not that one. It's about 10 seconds long, but here's another representation by a gentleman named Drew Barry. I love his work. I could sit and look at his work all day. What's striking to me is how fast, right now in your body, you're transcribing this DNA to RNA to make your proteins with all those free bases floating around. And I use the word base to describe every single one of those ACTGs. So that's what it looks like at your speed right now. So how does this impact computer scientists or data software engineers? Well, if it's an ATCG, we can represent it as strings. In fact, much of bioinformatics is string manipulation. So if you work on a project, you're most likely going to have strings. A lot of strings, really long strings. And we can use strings to represent DNA, RNA, or amino acids. And you can see here on the screen how DNA has A, C, T, and G, RNA has the A, C, U, and G, and then amino acids use letters to designate the different amino acids. And I believe there's like 23, maybe 25 amino acids that are in the human body living that make up you. So it's fairly finite set. So the question is, what can you do with these strings? Well, you certainly can compare them. And I'm a natural language processor, NLP engineer and an NLP tech lead. So we do things like the Hemming distance to compare two strings. We also combine them to make bigger strings. We cut them apart to make substrings. We reverse them. We do a lot of wonderful things with strings, and that's what we'll be doing today. So you might be asking, well, where do I get these strings? On the top left, that's an Illumina machine. That's a short-read sequencer. So they take the biomaterial, they pre-process it, they run it through the machine, and they amplify it and they multiply it. They take a really long piece of your DNA, or a DNA, not necessarily yours, break it into little pieces, and they multiply it, and then out comes the results. That's called short-read sequencing. That leads to a challenge on the post-processing side, because now you have all these small pieces that you have to recombine back together to figure out what you're doing. The advantage, it's really fast. The Human Genome Project, this is called next-gen sequencing. I don't know if they started with next-gen or they evolved into it, but that's the way they started evaluating our humans' DNA as well as other living animals, organisms. In the middle, that's Oxford Nanopore. That's a long-read sequence. They have a little hole, and they string the DNA, they cut it apart, and they string one line through it, and it reads each time. The advantage, you can make really long strings, so you have a much higher accuracy. The disadvantage, it's slow. So a lot of companies actually combine both short-read and long-read sequencing. In the bottom right is the kind of machine that I use. That's a mass spectrometer. That's what went off the rails in Half-Life video game. We take proteins, we take some biosamples, we break it down in the wet lab, and then we send it through, and then we get these strings back. Again, it's much like the short-read sequencing, we then have to figure out how to reassemble what we just saw. So when we build these strings on the short-read sequencing and then on the mass spectrometer, there's a couple different ways we build strings. We do a de novo sequencing, which is we take them all together and we try to figure them out, put them together like a really big crossword puzzle. It's actually more like a jigsaw puzzle that you put together in the dark, in a closet, with a lot of the pieces multiplied that you don't need. And there's no edge pieces. That's de novo sequencing. Some really cool algorithms to help along that. Secondarily, once you have a reference assembly, a dictionary approach, you have all these pieces, you apply them to the dictionary, and you try to extend out and saying, do I match, do I match, do I match? Constantly going back to the dictionary as you extend out the strings. A lot of people do hybrid sequencing too. So real quick, oh, this does not show, So apparently when I'm not, so I've had some problems with PowerPoint. I know, I'm the first one ever. I'm going to try to run this from here. Slideshow, play from current slide. Let me see, can you see that okay? Yeah, so I'm going to give you 5 pieces of DNA that came out of maybe a short read sequencer, and I'm going to help you and say that there's no duplication. How would you assemble that to make a single string? I will tell you that 2 out of the 3 letters are overlapping. And you can see which of the 5 pieces doesn't have an overlap on the right 2 sides of all the 4 others. And that would be the, I believe, I don't know what just happened. Oh, goodness. I can't make fun of Microsoft in these presentations, right? 1 second while I rebuild this. Oh, no. All right, let's try this. I don't know how many times I've practiced this, and that never happened. All right, so let's see if this happens. Okay, so we have TAA. We know it's the beginning one because there's no other of the remaining 4, they don't end with TA. And then we can take the first one that starts with AA, and then the next one that starts with AT, TG, then GT. And so we know our string, that's the full amount of our string. So that's the way we piece these things, these jigsaw pieces together in the dark. We can also do a dictionary where we have a reference genome. And then we just say, okay, where does AT fit into this? Then ATG, et cetera. You just sort of match it all together, and then you get your results. What I do with Hype Search on the mass spectrometry data is we do something called seed and extend, where we take, we have a reference genome, and we take the first fragment, then we take the next fragment, and we see if we can get some overlaps. So we've now gone from 3 to 4, and then to 5 to 6 to 7. And then we also have pieces that are laying around that don't fit the reference genome at all, or reference assembly at all. What does that mean? Well, actually, we're really interested in that. And then we turn around and try to do de novo on that, because that's what we think might be the cause of type 1 diabetes. Or actually, and it could very well be any autoimmune system, where the body is generating something that it doesn't recognize, and so it's attacking the area that's generating those particular peptides. So that's seed and extend. The lab and all the academic articles I've read, all the people I've worked with, when they look at code, code is something that gets in their way. They're PhD types, they're biologists who are learning to code. They could really benefit from all of your experience as software engineers. They all do imperative code. I'm trying to convince them maybe functional is actually a better way of thinking about it. I'm hoping to convince you. But think of a lot of loops. Think of a lot of mutation. Think of a lot of spaghetti code. The code base quickly goes like this. It fans out, and so it becomes very hard to maintain. When I came into the lab, that was one of the things we talked about, is how we can move from functional imperatives code to functional code. Now, some of you are probably C# developers. It's a .NET predominant conference. Love C#, but any effort I've seen to try to do object-oriented principles, design patterns, inheritance, all those sort of things, don't really work with this kind of scientific computing, so please, it's fun to do, but I would say look more at imperative and functional style in C# if you're bound to determine. Most labs use Python and/or R, and then maybe some scripting languages. So I presented this paper, or this presentation at NDC, and I said we're going to do the read alignment problem. I, in fact, switched it to the origin of replication problem because I think it's a little bit more accessible. In addition, if you remember that book that I referenced in the beginning, they made that chapter 1. So I'm like, oh, so if you read the book, you can see the code that they wrote and sort of their ideas, and then we can see it from a functional point of view. The origin of replication problem is this. You all are made of cells. I'm made of cells too. Cells die all the time. We need to regrow those cells. So cell division is very important. What happens is you have a single cell inside the nucleus. Remember, you have that DNA. It uncoils. It gets cut in a certain place. When it gets cut, another enzyme comes in and then splits it apart and then creates another set of DNA from the template. So it cuts it and works in 2 directions for 2 strands. We now have 4 strands. They go out and become the 2 daughter cells with the 2 nucleuses, with the complete copy. Most of the time, it's a complete exact copy. We do get mutations, though, when it does that calculation or that process. So that's something to think about. But I wanted to show you just another quick video, if I can. So this is really cool. Again, this is, what's his name, Drew Barry. And I could sit and look at this all day. One important thing, and for our just coding in a couple minutes, is that we read DNA from one end to the other end, the 5' end to the 3' end. I believe the 5 and the 3 represent the number of free carbon atoms sticking off the end of it. But we can only read in one direction. So when we split it, one's 5 to 3, one's 3 to 5. Well, it's easy reading 5 to 3. You just have to match on like it's doing down there in the bottom. The top one, that's 3 to 5, has to go up a little bit. You can see that green thing that sort of circles it around, writes it back, and then does it again and again and again. And it does 4 billion base pairs. I don't know how long it takes, not that long at all, to divide a cell, a human cell. So you can imagine the speed on which it's doing this. It's pretty amazing. All right. So that split, when it first cuts the DNA, is really important. That's called the origin of replication, or ORI. And the reason why it's important is if we are going to do any modification of the genome, or if genomes modify, if it modifies in there, inside that area, well, then the body doesn't recognize to cut it there. And so if you make any modification, let's say you do a point mutation that cures cancer, hypothetically. If you go in there and you modify in that box, that DNA box, well, that one cell is now cancer resistant, but it cannot propagate, and it cannot go throughout the body. So you've just solved one problem, but you've introduced another. So it's really important for scientists to understand where this happens. It's been a problem for them for quite some time. So they did a brute force mechanism. As an NLP engineer, I would love to think that DNA is color-coded. It's not. I would love to think that it uses a dictionary or a different set of letters to say, yeah, this is the origin of replication. It does not. We have absolutely no idea. When we started, we had absolutely no idea where the body said, okay, this is where you to cut. How did it know where to cut exactly? So scientists, like I said, did brute force. They took a smaller organism. So instead of the 4 billion base pairs in a human, they might have started with a simple virus or simple bacteria, maybe 1 million total base pairs, maybe even 100,000 base pairs in the entire genome, and they started knocking it out. They found a piece of it, and they cut it out. Now, this is sort of a brute force method. CRISPR-Cas9, obviously, we can be much more sophisticated now, but work with me here. This is the problem they had to solve. So scientists went through a very simple organism, and they started cutting out pieces of the DNA, and they said, okay, when did replication stop? So they started there, then maybe there, then maybe there. So we are going to look at cholera, which is only 1.1 million base pairs, and that box, the scientists determined, was about 300 base pairs long. And the next thought is, okay, so in a simple organism, this is the way the box is. Is there something in there that we can then use to extrapolate the box in a human that has 5 billion base pairs versus trying to knock out, you know, certain pieces of 5 billion? That's very expensive and time-consuming. That's where a bioinformatician can come and help find a needle in a haystack. So this is the example that Computational and Peisner used. This is an Edgar Allan Poe story called The Gold Bug. And I'm giving you a cipher. It's a bunch of letters that represent, or characters that represent other letters. We'll assume that it's English. What's the most common letter in the English language used? E. And that would be, then, making that assumption, an 8. What's the most common word used in the English language? That's right. So let's assume that semicolon is a T, 4 is an H, and 8 is an E. And then from there, we can sort of build out this Rosetta Stone to understand what that message was. Now, we're not going to do that for this message, but we are going to do it with DNA from cholera and see if we can apply it to the human body. So yeah, it's sort of like Wheel of Fortune. The scientists told us, okay, in cholera, that 300 base pair box, that's what we know. Can you tell us, bioinformatician, anything special about this? Can you derive a dictionary? There's only 4 characters. There's no white spaces. And actually, as a NLP engineer, I was really excited when I first came to this. I'm like, this is cool because I can do this stuff, right? I have all these cool dictionaries. No, I don't. None of the dictionaries that I know of could really help because there's no white space. You have no idea how many characters each word might be to then identify, what kind of patterns you see. So I think bioinformatics has it hard. Just as an example, You just read bioinformatics had it hard, and you thought, okay, there's 4 words or 4 tokens in that sentence. Well, it turns out, if I split it another way, there's actually 6 perfectly valid words, actually 5 perfectly valid words, and attic might be misspelled, and there's always error in our DNA when we read it out, so it's kind of a comparable thing. So a little bit harder. So I'm going to give you a little bit of dictionary, and we're going to get into code, I promise. When we say words in a sentence, those also sometimes NLP engineers talk about tokens. We're going to call those k-mers. And if the word, like the, is 3 characters long, we're going to call that a 3-mer. If a word like here, 4 characters long, we're going to call that a 4-mer. If the word is 2, we're going to call it a 2-mer. Yeah. So in this example, you can see I have a 6-character string, and I'm going to say, okay, we're going to start with a k-mer, or a 2-mer, and what is the most frequent word? And in this case, it would be AA, because if you go here, there's an AA, then there's another AA, there's another AA, and then there's an AT and a TG. Okay? So let's do this for reals. When we start pattern counting, this is the kind of code that you will see all over the place. On the left is the pseudocode. On the right is perfectly valid Python. I'm not going to tell you if there's an error in the logic. But you can see there's mutability, there's for eaches, there's if statements. I don't know if anyone's familiar with the expression triangle of doom or pyramid of doom, when you have so many if-nested statements that your code looks like a huge pyramid going the wrong way. That's what you see a lot of, and that's what I'm trying to avoid by using a functional style in F#. So we're going to keep this model in our head, and then we're going to see how F# solves these problems. And to do that, we're going to learn some F#. Does anyone use F# besides me? Oh, okay, cool. Great, great, great. The rest of you, I'm going to give you a 5-minute introduction to F#. Any F-sharpers, feel free to interrupt or expound on what I'm going to say. I am going to get out of here, and I'm going to Visual Studio Code. So just understand, if you're not familiar with F#, that everything is either data or function. There's no design patterns, there's no curly braces, there are no return statements, there's none of this boilerplate code, decoration code. It's just, if it's data, great. If it's not data, it's a function. If it's a function, great. If it's not a function, it's data. That's it. Every function is defined with a let if it's named, and white space is important, much like Python. So if you're a curly brace or a JavaScript person, you do have to understand that where the white spaces go into your functions, that is important. And the last line of every F# function is the return value. There's no return keyword. So if you look at lines 6, 7, and 8, excuse me, and people can see that okay, I hope? Okay, good, I'm seeing nodding heads. That is a function called add1. Add1 takes in a single parameter. I'm calling it x. Notice I'm not giving it a type because the computer and you are smart enough to figure out that it's an integer based on your usage inside the function. And I'm adding 1 to it. So I'm going to send this down to the REPL, read, evaluate, print loop. If you came to my talk 6 years ago at NDC Oslo, you would have seen me demonstrate what the REPL does in all its glory. But for now, I'm just going to use it. And you can see at the bottom, it creates a function named add1, takes an x, which is an integer, and returns an integer. So if I send in 10 to add1, I get 11. Send in to add1, I add 3. I'm going to get 4. The next piece is I'm going to create a sequence of 1 to 10. Okay? And then I'm going to apply a function to each item of that sequence of 1 to 10. That's what seek.map does. You have 10 items. It's just going to go to each one and apply the same function. And the item that you have in that original sequence of 10 is the input parameter. Over here on the right is another function. So seek.map is actually something called a high-ordered function, where we pass a function into the function. When I came from C# to F#, that was really hard for me to wrap my head around. I'm like, no, data goes into a function. And F# says, yeah, data can go into a function. But honestly, you can pass a function into it. And so whatever function you pass into it, I'm going to then execute for you. So in this case, I passed in an anonymous function, a lambda, defined by that fun keyword. Fun takes a single value, x, and then adds 1 to it. It's exactly the same thing that add1 does up here on line 7. And then when I do seek map, it's going to apply to each of the items on that list. 2, 3, 4, 5. If you want, I can do something like add 10 to that. 11, 12, 13. So if you've gotten this far, you're done. Everything else is easy. And then we can go to the DNA, I promise you. When I took that list of 1 to 10, I can also do that seek.map, but instead of doing a lambda, I'm going to add 1 to it again. I'm just going to call a named function, and it does exactly the same thing. You don't have to pass in parameters or anything. It already knows. Why we love F# is that we can then chain functions together. PowerShell folks probably are very familiar with this too. You take a function, you run it, you then take the results and you apply it to another function, and then another function. You just keep piping along. You can run each one independently. And so I can take my list of 1 to 10. I am then going to add 1 like I did before, so 2, 3, 4, 5. I'm then going to say, okay, I only want the evens. And then I'm going to square it. And then I'm going to pass it to a final function, which is not a high-ordered function. It's just sum up everything in that list. So 220. Okay? I'm seeing some nodding heads. The F# people are familiar with this. The C# people might be a little bit out of sorts, but I guarantee you as we use this, it becomes very familiar and intuitive. What? Yes, sir?
The exclamation mark larger than, is that the piping of the data from the previous one?
Yes, that's right. That's this character right here. This thing right here, you mean? Yeah. Yep, it pipes. So here we have our list of 1 to 10, and we're going to apply another function called windowed, which I absolutely love for a lot of my bioinformatics work. This takes that sequence and just runs a window down it and combines them together. So we have an array or a list of a list. So that first list is 1 and 2, then 2 and 3, 3 and 4. I want to do a window maybe not of 2, but I want to do a 4. And so you can see 1, 2, 3, 4, 2, 3, 4, 5, et cetera, et cetera. Cool? That's all the F# you need to know. We are now ready to tackle biology. So if you remember, we did that pattern count code. And I'm not going to go back to it to refer to it, but this is what we're going to do. We need to count up how often a pattern happens within a string. The string might be 4 billion base pairs, but let's start more modestly of 20. So what we're going to do is we're going to make a function. Of course, we always make functions in F#. We're going to pass in the text, and then we're going to pass in the pattern. So if I have a text of 20 characters long of DNA, well, I know the alphabet's either A, C, T, or G. Let's say I want to know the pattern CAT, cat. Sorry, dog lovers. DNA only has cats. So we can count how many times the word cat shows up in this string. So we write that window going down. We then say, oh, I hate this, but when we do a windowed, you do a window to a string, it takes it and makes the results now an array of characters. So I'm going to bring it back together to make it a string. So I can then match it to the pattern down here. Hey, the pattern that I just sent in, does it match any of those windows? And if the answer is yes, tell me how many. So here's some text. How many times does cat show up? Anyone know? If I put 5,000 characters up there, you wouldn't know. But this function, or acat, can tell you pretty quickly that it's 2. Okay? So let's do this a little bit at scale. Let's make a dictionary of 1.1 million base pairs. Oh, I'm sorry, I'll make the string join again. Which is that, remember that cholera genome that we're going to look at? So there's 1.1 million length string. How many times does acat show up in there? 4,341 times. Okay with what the function does? Okay with how we built it? I'm seeing nodding heads, so we can move on. What I want to do now is show you the next piece. So we now can count how often something happens inside a string. We then want to do our word count to see what's the most popular word, because maybe the most popular sets of words in that box of the origin can give us some clues. So that's the way they presented the code. This is where I gave up and went straight to F#. I'm like, this is nonsense. This is 33 lines of code, and all you're doing is counting words. So let me show you how we do it in F#. Up here at the top, I have a function called frequent words. and I'll review it for you in a second. I just want to send it down to the REPL. We pass in the text, and we pass in the K. Remember that K-mer? So we can pass in 2, 3, 4, 5. Show me for maybe all words that are 5 characters long, what's the most popular one in that group of, in that sequence of DNAs. So I'm putting a function inside a function, a subfunction. We're going ahead and using our window friend again. We're then going to map it to a string because they were arrays of characters. We're then going to use another function called countBy that just says, okay, count the number of occurrences we see. It's very much like a group by if you're familiar with SQL, or if you're using LINQ, right? So I just want to count by the ID, and then I want to flip it around and sort it by descending. There is a data structure, much like in Python, called a tuple, or a tuple, depending on what part of the world you're from. And so when I do the count by, it says, okay, AAA, 4, CAT, 6, TTT, 3, right? It's paired. So all I'm doing when I do the sort by descending, I say, okay, give me the highest number one and put me up there. And then I take the first one, the head of that list, and I get that number. Oh, sorry. Did I not? Oh, yeah, I know why. And then I'm sorry, I shouldn't run that. It's a subfunction. I then find the most popular number, and then I say, okay, go back and find in that groupings anyone with that same number. So in case we have a tie for the most common combinations, we can handle that. So that's 5 to 14. That's 9 lines of code. I find it very intuitive when I read it, how it's handled, and I can throw away 33 lines of Python code, which makes me feel really good. So I am going to take the origin. Remember before how they did the knockout to identify it for cholera? I'm going to take that, and I'm going to stick it into a value called ORI, and it's 541 base pairs long. I am then going to make a list, 3 to 9, and that'll be my K, and I'm going ahead and I'm going to call frequent words to find out the most frequent words for this. If you can see that on the bottom, I hope you can. The most frequent word for a 3-mer is TGA. I guess maybe not surprisingly. The next, for a 4-mer, it's ATGA, so they just stuck an A on it. Then surprisingly, for a 5-mer, it doesn't build off that TGA. And you can see the counts as it descends, which intuitively makes sense. The smaller the word, the more likely it's going to show up in those 540 or 541. So TGA shows up 21 times, and then 12 times. Anyone see anything interesting about this list as you go down it? Any analysts out there? Or people who like to do crossword puzzles or finding commonalities or outliers? The answer is 9. If you notice this is not a linear descent, it's actually a log. 9 should probably show up once, if once. Instead, we're showing 4 different patterns. And they're showing up 3 times each. That's a lot. So what's up with this 9-mer? Is there something more we should investigate? And the answer is yes. So what we can do, there's that code again, is I want to show you, yeah, I can show you from here, something called the reverse complement, which in America, when you give someone a reverse complement, you're actually insulting them in using complimentary words. As an NLP person, I love it. But remember, we unfolded that DNA, we unwound it, and it got cut apart into 2 strands. I've just shown you 1 strand. But there's another strand, right? The reverse and the complement. So for this example, A, T, G, C, G, etc., it has a reverse complement, and you can see it underneath. So going back to that first 9-mer that we thought was interesting, we come back to the end, the far right, and we say G. Well, remember, its complementary pair for a G is always a C. That'll be the first position. Then the A, its complementary pair, a T. That'll be in the second position, et cetera, and we build it out. And there's that pesky cat again. Just if you do some basic statistics, remember I did that random of 1.1 million. The chances of a 9-mer showing up 3 times in that 500 base pair box is about 1 in 1,300. But we actually have a reverse complement, so it's showing up 6 times. And that's 1 divided by a lot. I can't remember the number. It's off the charts. So let's go take a look at that and see if there's something in the code that'll show us of those 4 NIMers, which one, oh, good, this one crashed too. 1 second, I'm sorry. Microsoft. All right, good, that didn't crash. So here's the reverse complement, and I should show you one more thing. Yeah, okay, thank you. This is the code for the reverse complement. The pseudocode makes sense. You reverse the string, and then you take the complementary base pair. So I'm going to go back here, and so it was showing, but I don't know what it's doing. Restart and updates. What in the world? And now it's trying to start Visual Studio, which, does anyone use Visual Studio? Yeah, a couple still. All right, we're going to stay in VS Code. So finding the reverse complement, I think it's fairly straightforward. We come up here and we make a function for it. And we take this character array, and I do a little bit of protective coding. I make it all lower invariant, and I turn it to a character array. Then I go ahead and map it. So this is actually complement reverse. So I reverse the reverse complement. So if it's an A, make it a T. If it's a C, make it a G, etc. Then I do seq.reverse. I turn it back into an array. So the reverse complement of ATG, of course, is our cat again. So let's do that frequent words now. And we're going to throw in not just reading it from 5 to 3 this end. We're going to read it from the other end back 5 to 3 and see how many we get. Make this a little bit bigger. So let's start with the 3mer. TGA shows up again, the 25 times. Its reverse complement is TCA, and it shows up 17 times. So that, on the strand, is 42 times. Same thing with 4, 5, 6, 7, 8, and 9. Check out this NIMer now. This and this are reverse complements to each other, and they're both showing up 3 times. So it'll, remember that 6 divided by 1 divided by, the chances of a NIMer showing up 6 times in a 500 base pair spring is astronomical. That's what we're seeing. We're also, we can throw out the other one because it's 3 and 1. So there's only 4. That's, again, a big number. But if we're looking for the THE, back to our cipher, we want the most popular word. And in the case, this NIMer that's showing up 6 times, that's it. Okay? So now let's see what we can do about this. Visual Studio. Quit. No, is PowerPoint dead on me? Oh, no, good. Whew. So we now think, okay, we have this NIMer that's this long. A NIMer that's, we have a set of base pairs that are 9 long. And we see it pretty frequently in this box of 500. But how often does it show up in the full 1.1 million the genome of the cholera? And they called it clump finding. That's fine. But basically, we're going to go through all 1.1 million base pairs with that NIMer and each section and see how often it shows up. And we have to assume that we don't know that it's 541, the box, or we'll assume it's roughly that, so we'll just divide it up into 500 groups to see, okay, if this origin of replication of 500 base pairs was in the first 0 through 499, how often does it show up? And we can just keep it going down like that. That's the intention behind the clump finding. So, make sure I'm okay on time, good. So what we're going to do is that pattern count again, same code as before. It's the same text, and you'll get the same result with our cat showing up twice. We're then going to basically take substrings to see if that substring appears or not. And if it does, return to me. This is going to say at index 12 for a 3mer, what do you get? In this case, cat. There's a really cool feature of F# called the option type. Right here on line 20, you can see that I return sum. If I have an index out of range kind of problem, I just return none. There's no concept really of null. I mean, we have to have null in F# to deal with the rest of the .NET ecosystem, but we really don't use it. So it just cuts down the amount of errors significantly when we start using sum and none types. And you can think of it as a wrapper. Like the Amazon box with the cool thing inside, the box will say there's something inside. You open it up. If the box comes and it says none, well, you don't have to open it up because there's nothing in there. So that's our way we're going to find that certain clump. And then we're going to pass in the text, and we're going to break it apart. So in this case, remember that I had about 20 characters. I broke it into groups of 3, substrings of 3. I can then go through those substrings and say, oh, is anything in there? And so remember, I broke that up into 3. This will say, OK, I'm looking for cat over here. How often does cat show up? And more importantly, where does it show up? So cat shows up twice. It shows up in index or number 4, 0, 1, 2, 3, 4. Yep. Where is that? CAT. I'm sorry, it shows up in 7. It shows up in clump number 4. 0, 1, 2, 3, 4, and it shows up in clump number 7. But we can search and make that clump even bigger and then search within it. So I made the clump the full length of that, and if I run it, it's going to show up in position. It's going to show up in the only 1, 0, and there's 2 examples of it. So not only can I look for clumps, but I can look inside the clump to find my pattern. In this case, the NIMER. So I'm going to pull in the entire genome. I'm going to assume that the origin of replication is about 500 characters long. And I'm going to go ahead and break those into 500. So 1.1 million divided by 500 is 2,000 something. We'll see in a second. And I'm going to then, remember, this was the first side, right? The ATG one. The cat's on the other side coming back. So we're just going to look for this one to start. We're going to search the clumps. And, and see what we get. So 500 into 1.1 million is 2,217. How many of those had 0 of that example? And the answer is 2,201. So this pattern is not appearing in the entire genome. It's only appearing in 16 of the 2,000 boxes. And so let's take a look at those. And you can see here at index 304, the 304th clump, so you multiply it by 500, you know exactly the area on the genome. There was 2 examples of it, and then a couple 1s. And since I sorted it by descending, we know the most common place is number 304. So let's take the reverse complement of it and do the same thing. It didn't run. 1 second, please. .304 again, and there's 3 of them. Why I don't think 6 showed up versus 5 is because it was actually 500 and something was the origin of replication, and 1 might have run over to the end, so when we divided by 500. But here I am, as a bioinformatician, going to the biologist saying, hey, your origin of replication, don't run 2,200 experiments to knock out each one to see where it goes. Go to 304 times 500, whatever number that is, knock out that 500 and see if the cell doesn't replicate. So we have just found the needle in the haystack for the biologists. Pretty cool, huh? We've now searched a genome of an entire, well, of a living creature, or it's a bacteria, and we found where it replicates, or where it divides to replicate. But there's more. The cool part of F# is I hate dealing with strings, because I think strings can be fragile to work with. You always have to process the strings, you have to check the invariant, the culture of it, et cetera, et cetera. F# has a wonderful type system. There's a good book called Domain Modeling Made Functional by Scott Voslin. Pick that one up too if you're interested. I am going to now make a type, A, C, T, and G. That's the entire universe of what it can be. And I'm going to do that reverse complement again, but I'm just going to turn it from strings into an array of DNA. Notice the amount of code drops significantly. In addition, if a string came in with the letter G, what's a good letter? R, it wouldn't hit the type system, so the compiler would get complained about it. So it's pretty cool. So highly recommend using the type system to your advantage and looking at that book. There's not a lot of like object-oriented programming, I notice there's a lot of stuff that goes on, and when you start using the type system, there's very little, but it's enough to sort of make sure that your code is right. Don't need unit tests either that way. One other thing we do is we know that the data comes in is sometimes there's differences between reality and when the mass spectrometer or the Oxford nanospore, maybe not the Oxford nanospore, the Illumina machine, throws some errors at you. So usually we don't just look at an exact match, we look at a close enough match. So this is called the Hamming distance. So if I take one former, like ACAT, and then I take another one called GCAT, and I ask what the Hamming distance is, it's going to say 3, and we get some. If there was no match, it would be none, or if the data was malformed. The code itself, I think, is fairly straightforward. If you've been staying with me, you basically check to see you've gotten 2 arrays of strings coming, or you have 2 strings coming in, you check their length. If they're not the same length, you throw it away and say, I can't do a Hamming distance. If they are the same, you make them both character arrays and you zip them together, just like this. And then you filter to see if the one side matches the other side. And then just tell me how many, where the match happens. So that's really handy, because when we do the pattern counts, most of the time, we're not looking for an exact match. We're looking for a close enough match, 90% match. And with the Hamming distance, you can just say, OK, what's my distance? And then what's the total? And then is 75%, 3 or 4, for example, good enough? And that's really for the biologist to decide. So there's some pattern counts. Same kind of thing here. We're going to look for that same NIMER again in the entire 1.1 million. But this time, I want to do the Hamming distance of all 9. Right? There's 8 of them total. Remember, 6 were in the box, or 5 were in the box, and 3 were spread out. And then we can also say, OK, but what about 8? Well, now we have 224 examples. So if we didn't get the box immediately identified, we might have to go up into 224, excuse me. And if we want to be off by 2 letters, for example, there's 2,420 of them. So that's what we use the Hamming distance for, and use it pretty frequently. One last thing I want to talk about, and I think I have a couple minutes left. Yeah, I've got a couple. I want to talk about another great thing of F#. And it's a library out there called Parallel. So when you start dealing with large volumes of string, it can be pretty resource intensive, compute intensive, big data. So I don't like writing code that just supports using more than 1 core on my machine. I'd rather it just does it automatically. And F# does it pretty much automatically. So here we are. I'm going to make, how many is that? That's 10 million. I'm making a string of 10 million letters, base pairs. And you can tell it's taking a little bit. The machine's complaining about it. We're still clocking. And then I'm going to send in our pattern count again. And let's give it a second to generate those 10 million. And I wonder if I'm presenting it. It should usually take about 10 seconds. In fact, it's taking longer. It's concerning. There we go. So I'm going to turn on the timer. Remember our pattern again. And remember, this is just an arbitrary 10 million base pair string. I'm going to then run it to see how long it takes. And it should take 8 seconds. Yeah, 8.7 seconds. I'm going to copy it down here. And then I'm going to go up here. Oh, I'm sorry. I ruined my aha because I didn't change it from the last time. That's the way it normally should run, and we'll see how long it takes. Again, it should take a little bit longer than 8 seconds. Yeah, 8.9 seconds. Depending on how you run it and how the single processor, because there's always a context switch between the processors. But my point is that to change it from using 1 core, which is what we've been doing so far, to all the cores that are available to me, I add a letter. There's no other code out there that I need to worry about. There's no mutexes or things like that. I just use pseek, which means parallel seek. So it's a really effective way, particularly when we use larger strings, to plow through it quickly. And so that's I love it because I get the speed without having to write the code. I can write the code that actually is more algorithmic than just trying to make things faster. So let me come down to here. So why do I think F# is better? I think it's a great language because you don't have code for code's sake, so it has a high noise to signal ratio. And it's very easy to sit down with a biologist and talk in F# to them, particularly when you use the type system. And that error reduction I talked about earlier. A big problem in academia is that reproducibility of research. You pick up a journal of XYZ, journal of bioinformatics, let's say. Well, bioinformatics actually came to solve the problem. A journal of biology, open it up, and there's an article that does some kind of analytical work. Most recent research, I think, says that about 30% of those articles, no one can figure out how to reproduce it. Think about that the next time you get your COVID shot, I think. But it's true that the reason that software engineers and functional languages can help here is a lot of the code itself, because of immutability, a couple other things, and also good software engineering techniques like source control, things like that, we can make experiments more reproducible, which I think will really benefit everyone in the long run. So that's what I have for you today. Want to mention that, like I said, I got into curing type 1 diabetes through the lab because it affected my family personally. Some of you might be affected, or families affected by other diseases like cancer, and you're interested and want to use your skills. Well, I'm not telling you to leave your 9-to-5 job. That'd be cool. But if you are interested in your 5-to-9 job, and you're interested in trying to help out the world, make it a little better, please contact me. I can tell you how to sort of get involved. There's different research groups out there that really need software engineering help, and it's a lot of fun. So if you really like building algorithms, I showed you a fairly simple algorithm today. It's a fantastic way to sort of help and give back. And it's pretty cool. Like, you know, I'm at a dinner party and people say, hey, what do you do? And I'm like, oh, you know, I build a phone app. Hey, I'm trying to cure cancer. Well, then they want to talk to you. So, you know, there's that social thing too. All this code is available for you on GitHub. You can take a picture of it there. And again, come out, talk to me, email me, tweet me, and I'm happy to talk to you about that or coffee farming. Thank you.